I'm here to present on the topic structure of hemoglobin. To start with introduction, hemoglobin is a protein which is a heme protein, and the types of protein are globular protein, which is a functional protein, and linear protein, which is a structural protein. And this hemoglobin is considered as globular protein, where mature RBC do not synthesize hemoglobin, where immature RBC synthesis hemoglobin, and mitochondria plays a vital role for the synthesis of heme. And hemoglobin is equal to heme plus globin. We all know that globin is a protein, and globin is considered as a protein so that it is synthesized by rough endoplasmic reticulum. It is a cell organelles present in the cells. And coming to the heme synthesis, heme is equal to protoporphyrin, that is protons plus ions. Protoporphyrin is equal to ion free heme. That is protoporphyrin consists of 4 pyrrole rings. Pyrrole ring is nothing but the ring which is present in the hemoglobin are said to be pyrrole rings, and the ions have 6 coordination. 4 bind with these pyrrole rings of protoporphyrin, 1 bind with histidine, which is an amino acid from globulin, and 1 is free, which is bind to oxygen and CO2 respectively. This slide shows about the structure of heme, where Fe is present at the center, and surrounding this Fe there are nitrogen, and 4 sides or 4 edges is connected with MVP, that is methyl, vinyl and propanol. So this is the full structure of the hemoglobin. It is a porphyrin ring, we will be saying it as. And next, this picture depicts the where this heme structure is formed. So it is formed from RBC, that is red blood cells. Next, it is forming to 3D structure, that is hemoglobin molecule, and from that hemoglobin molecule of 3D structure, which consists of subunits, along with the heme, which is present in between the folded structure. Finally, the heme structure is obtained, and each hemoglobin molecule, which is a globular protein, consists of 4 heme, 4 linear protein, each one bind to a heme. And heme synthesis starts with 2 simple products, that is glycine, which is an amino acid as we all know, and succinyl-CoA, which is an intermediate of citric acid cycle. This citric acid cycle is a carbohydrate metabolism, we have read it before. Then second one will be the reaction of these 2 simple molecules together with a cofactor, that is vitamin B6. It is a nothing but pyridoxine, which is useful for brain development, and it will produce amino-levulinic acid synthase, that is simply in it is called as ALA synthase, and this step also called regulatory step or rapid limited step. And regulatory step, when this step being enhanced, all of the pathways will be enhanced, that is in a fast manner, and if this step have inhibitory action, all of the pathways will be inhibited or terminated. Then some steps will happen in the cytoplasm that is not necessary for the heme productions, and then this molecule will return to the mitochondria when a protoporphyrin is formed, and this protoporphyrin will bind to ion, which is a ferrous. At the same time, globulin is being synthesized in rough endoplasmic reticulum, and protoporphyrin plus ferrous plus globulin will form a hemoglobin. So the complete structure of hemoglobin comprises of protoporphyrin, ferrous and globulin. Then this slide shows about the ALA synthase, that is amino-levulinic acid synthase, which is formed from which acts as a enzyme for the conversion of succinyl-CoA and glycine to amino-levulinic acid from the area of mitochondria to cytosol. So once this formation is formed, that is the amino-levulinic acid, this in turn converts to phosphorbilinogen, that is PBG. Short form is and with the help of the enzyme ALA, that is ALA dehydratase, and once this phosphorylinogen is formed, it again forms urophosphorylinogen 1 with the help of the enzyme HMB synthase, that is urophosphorylinogen 1 synthase. And from this urophosphorylinogen 1, it is converted to urophosphorylinogen 3 with the enzyme urophosphorylinogen 3 synthase. And from the urophosphorylinogen 3, it converts to coprophosphorylinogen 3 with the enzyme urophosphorylinogen decarboxylase. And this end product coprophosphorylinogen 3 enters into the mitochondria as protophosphorylinogen 9 with the help of the enzyme coprophosphorylinogen oxidase. And the product which enters as protophosphorylinogen 9 is converted to protophosphirin 9. First, it forms from protophosphirin 9 to protophosphirin 1, and it is converted to heme with the addition of Fe2 plus and ferrochelatase. Okay, what is the enzyme? Ferrochelatase with the Fe2 plus entering into N to form a heme. Along with this heme and glucose will inhibit the synthesis of ALA synthase. Okay, which will inhibit the synthesis of ALA synthase, whereas alcohol barbiturate hypoxia will stimulate the ALS synthase for the production of amino linoleic acid. So this process occurs in the mitochondria. And ion in hemoglobin is ferrous or ferric. Why? What is the reason? So ion in ferrous form, it is of 6 coordinate. 4 bind with the protophosphirin, as I have mentioned you earlier, and 1 bind to histidine, which is a amino acid, and 1 freely bind to oxygen or CO2 respectively. Wherein ferric form, we will have 1 bond missing, that is small functional hemoglobin. So heme and globulin are not isolated and bind together by ion. So when oxygen binds to ion, this process is called oxygenation, not oxidation. Okay, and oxygen transport through circulation as a molecular form, it is O2. This oxidation stage of the ion does not change. The oxidation of Fe2 plus to Fe3 plus only occurs occasionally. The oxidized form methemoglobin is then no longer able to bind O2. The proportion of methemoglobin is kept low by reduction to only 1 to 2%. And the types of hemoglobin is normal or not. Different types of hemoglobin due to the different type of globin. It is not a heme. Adults have hemoglobin A and small amount of hemoglobin A2, and fetus have hemoglobin F. So these are the types of hemoglobin, where hemoglobin A has the formula of alpha 2, beta 2. These are the subunits I have mentioned you earlier in previous slides, and HbA2 is a alpha 2 and gamma 2, and HbF is a alpha 2 and gamma 2. Again, in HbA2 it is alpha 2 and epsilon 2, and in HbF have more affinity to oxygen than HbA, because it has a subunit of alpha and beta. Here the subunit is, the radiation is very high, that is alpha and gamma. So gas transportation. Why we use hemoglobin as a transporter for oxygen? Because of low solubility of oxygen in water, hemoglobin have a regulatory effect on oxygen concentration in lungs and tissues. All tissues required for oxygen and 20% of CO2 from metabolic waste transport via hemoglobin. An allosteric effect. When an enzyme reacts to effectors with the conformational changes that increase or reduce its activity, it is said to show allosteric behavior. That is, if there is any enzymatic changes, it is said to be allosteric effect, and many substances act on hemoglobin as an allosteric substances. Although hemoglobin is not an enzyme, but it have all the characteristic of enzymatic reactions. Then substances that have an allosteric effect on hemoglobin are 2,3-DGP, that is diphosphoglyceric acid, and intermediate factor in glycolysis. And second one will be proton, that is pH, which is effect on hemoglobin, and third one will be carbon dioxide. And how this hemoglobin is formed? When hemoglobin have low affinity to oxygen in tissue, it is called deoxyhemoglobin, and it is referred to as T form. And T is for tens. When hemoglobin have high affinity to oxygen in lung, it is called oxyhemoglobin, and it is generally referred to as R form, that is R for relax. So this slide shows about the oxyhemoglobin R form. Left hand side picture and right hand side picture depicts the deoxyhemoglobin T form. T stands for tens and R stands for relax. Here, the allosteric substances act as a stabilizer for T form of hemoglobin. In other words, affecting on the saturation of hemoglobin with oxygen. This picture depicts the partial pressure of arterial oxygen. It describes the relationship between the saturation of hemoglobin and the partial pressure of arterial oxygen. It makes sense that the more oxygen that's available, that is higher PO2, the more saturated hemoglobin will be in a percentage saturation. But what if the hemoglobin is in a different conformational state because of acidosis or a hemoglobinopathy? And once a hemoglobin molecule is saturated with oxygen, it readily gives up the oxygen to any organs and tissues that it is required for. The majority of adult hemoglobin is hemoglobin A, an iron-based metalloprotein made of 2 alpha and 2 beta globular protein subunits, that is alpha tetrameric. Each HbA molecule can hold up to 4 oxygen molecules, but the hemoglobin molecule itself is also involved in physiological buffering and carbon dioxide transport, that is carbaminohemoglobin. Then myoglobin saturation curve do not show allosteric effect because of no affinity to 2,3-DGP, that is diphosphoglyceric acid. Then hemoglobin saturation curve is sigmoidal, which is S-shaped, that is saturation changes due to allosteric effect. And steps for O2 and CO2 transport. In lungs, pressure of oxygen is high, so it diffuses from outside the body to inside the body and bind with hemoglobin. And in tissues, hemoglobin bind to 2,3-DPG, and that causes to decrease affinity of hemoglobin to oxygen and releasing oxygen to the tissues. In the same time, CO2 is being bind to hemoglobin due to high pressure of CO2 inside the tissues, and that reducing hemoglobin affinity to oxygen, which is a allosteric effect. An effect of allosteric effect is, and 2,3-DGP or BGP enters between two chains of beta and convert hemoglobin from relaxed form to tense form, reducing affinity of hemoglobin to oxygen. Then proton or a pH and CO2 formed from metallic or metabolic reactions in tissues bind to hemoglobin and decrease the affinity of hemoglobin to oxygen, and specifically called as Bohr's effect. And this slide shows about the hemoglobinopathies, which is a disease for a particular patient who is having this type of hemoglobinopathies. And these are the types of hemoglobin types, which is a HB part, HB HBA2, HBS, HBC, and HBSC. And for example, HBS is the hemoglobin type that will particularly prone for the disease sickle cell disease, and the features will be sickling of cells at low oxygen conditions, vaso-occlusive crisis, hemolysis due to distorted shape, etc. And hemoglobin SC, the alpha chains will be normal, and again this is from the disease of compound heterozygous form of sickle cells and HBC, and the features will be symptoms of HBC and HBS. So thank you.